Right, well, good morning, everyone. I hope that you can all hear me. I'll just get rid of those subtitles because we don't want anything getting in the way. So I can't see anything other than my slideshow, so I'm trusting that you can hear what I'm saying and that you can see the slides. Well, good morning. Thanks for coming to this masterclass. This presentation is really all about our MSc in Analytical Science for the Biosciences industries, and I thought what I would do to give you a flavour of the kind of work that we're doing in and around the MSc and also associated research projects is to talk a little bit about a single case study in the sphere of whisky science. And so without further ado, we'll crack on. Okay, so I've changed slides. Hope you can see the fact that I've changed the slide. Basically, the agenda's in 4 parts. I'm going to talk a little bit about Scotch whisky, a little bit about peat, and then the kind of problem that we have with peat at the moment and the manufacture of Scotch whisky, and how analytical science can help us to hopefully solve this problem. So I don't know how many of you out there are whisky drinkers, but if you're not, then there's still an awful lot of people who are. It's Scotland's most famous drink, with a potential exception of Iron Brew. It has an enormous export value to the UK of over 6 billion, and those figures are a couple of years old now, and they're still rising. It's a very traditional spirit. It has only 3 basic ingredients: water, yeast, and barley. And those ingredients and the processes by which whisky is made are very tightly regulated by the Scotch Whisky Association here in Scotland. And therefore there isn't an awful lot of wriggle room in terms of how you actually make whisky. It's very prescribed and very traditional. Some whiskies, but not all, are characterized by what are known as smoky flavors and aromas. So there are very distinct smoky taste when you taste the whiskey. And we now know that those smoky aromas come from one of the elements of the manufacturing process of whisky at the very beginning of the process, where peat is burned in a kiln, or smoked to be more accurate. It's not actually burnt. And the original reason for doing that was to dry the barley, which is one of the ingredients of Scotch whisky, prior to the manufacture of whisky spirit. And the reason that peat was used was because it was a very common fuel in Scotland, in the Highlands and Islands in particular, the Western Islands. And why not use it? Use it for heating your house. So if you needed to use it to heat something else, then why not use peat? So peat became used in the early distilleries, quite widespread scale, to dry the malt prior to the manufacture of the whisky. This is a kind of blurry cartoon, but it gives you an idea of how whisky is made. I want to go through all of this. I think I've got a little presenter here, slide laser pointer. There we go. It will take the whole webinar to go through the whole process. But if you go to the top line here, you'll see that one of the very early elements of the whisky making process is this element called kilning. And this is what I was referring to just now, where you take your barley, your cereal, spread it out on the floor, the malting floor. Underneath there will be some peat, and you'll smoke the peat, you'll cause it to smoulder, and that will dry the barley. And in doing so, the peat smoke, which will carry chemical compounds from the peat into the barley, and that's how these smoky flavors were originally introduced into the spirit, sorry. And as chemists, we understand what those compounds are, and this is a fairly bewildering slide. On the left here, you've got what we call the Scotch whisky flavour wheel, which describes all of the different flavour descriptors for various kinds of whisky, combinations of different flavours. And you'll see at the top here, around the top, what we've got, this peaty segment of the pie, and that leads to these flavour profiles which are described as burnt or smoky or medicinal, very distinct flavours for certain types of whisky. And on the right hand side of the slide, you can see that there's a number of chemical species that are drawn up, all based on phenol, so C6H5OH, so it's a benzene ring with a hydroxyl group off it, with various other substituents. And it's these phenols or phenolic compounds that are believed to create these smoky aromas and tastes in Scotch whisky. And so if we go back a step to the peat issue, so these are pictures of peatlands in Scotland, and we know now that these smoky notes, smoky flavors and aromas that arrive in the spirit come from peat smoke. But we have a problem here because, as many of you may know, it's kind of high profile in the media at the moment. Peat is a limited resource. It's running out in a number of ways. One is because it's been harvested as a fuel and indeed to make whiskey, although the amounts of peat that are used to make whiskey are relatively small, but also because of degradation, because of climate change, human intervention. And so peat is in crisis in the UK, and it's believed that something in the order of 80% of the UK peatland estate is damaged in some way. And what is even more critical is that we know that peat is a huge bulwark against climate change. It's a carbon sink, much more effective than forests, in fact, at storing carbon. And so the idea of losing large amounts of peat is just going to make the situation with regards to climate change even worse than it is at the moment. So there's a number of problems with peat, and that's led to government organisations to begin restricting the use of peat in a number of spaces, particularly in casual horticulture, for example, in the UK, in England and in Scotland also. It will no longer be legal to use peat in, for example, compost over the next few years. So gradually the rules are tightening over the use of peat. And so this might have implications for the Scottish whisky industry if there are regulations and restrictions on the use of peat, for example, in the production of Scotch. So what we're going to do in this project is to use analytical science to try and identify a substitute for peat in the production of Scotch. So what we want to do is to find something that's more sustainable than peat, renewable than peat, that can generate those same smoky flavours that we get in our Scotch whisky. And this may seem a little bit strange, a little bit out there, but one of the things that we've looked at, one of the materials that we've looked at is actually coffee waste, so spent coffee grounds, fancy title SCG. And this is attractive in a number of ways. One is that there's a huge amount of it. As this slide indicates, it's about 10 billion kilograms of coffee was consumed, and that's many years ago now. And so typically you generate about 6 million tons of coffee waste every year in the world. And of course that goes to landfill largely, although increasingly it's being recycled as a biomass for use in renewable energy, for example. And the other thing that's attractive about coffee waste is that although it seems a million miles away from peat, the actual biomass, the core materials, the backbone of coffee are these biopolymers, cellulose, hemicellulose and lignin. And these are the same biopolymers that are known to form the bulk of the peat biomass. And it's from the cracking, the thermal cracking of these biopolymers that we believe these small phenolic flavor compounds come. So it's interesting to speculate that if we were to smoke coffee waste in the same way that we smoke peat, we might produce those same phenolic compounds and generate the same kind of smoky flavors and aromas that we could from smoking peat. So how are we going to try and address this and see if our presumptions are correct? Well, we use analytical science. And I'm going to show you a few results, a few outputs over the next few slides. They look a little bit intimidating, maybe to some of you, but I think the point that I'm trying to make here is that we can use a range of analytical science techniques to gather lots of data from different sources to try and understand whether we could, if the situation arose, use spent coffee ground, for example, or any other material as a substitute for peat in the production of Scotch. So how will we do this? So one of the techniques that we can use is a technique, nuclear magnetic resonance spectroscopy. So NMR spectroscopy is a very common tool in analytical science and analytical chemistry, and also in bio sciences as well. And it's often used to characterize pure compounds. If you're working in a lab and you're making a chemical compound and you want to know what it is, or to prove to yourself that you've made what you think you've made, then NMR is the go-to technique to try and do this. And it's quite attractive in a number of ways. It's quite simple to do. Sample preparation is quite easy. Interpretation can be a little bit tricky, but there's a lot of data out there already that you can use, and with some experience, then you can usually get to an answer relatively quickly. The disadvantage of NMR is it's very expensive. NMR spectrometers cost about as much as a house, and therefore it's not widely applicable in a number of spaces, and it's not the kind of technique that the whiskey industry itself would use, for example, to analyze Scotch whiskey, because it's simply too expensive, it's too immobile, and it demands a great deal of maintenance and expertise to actually keep the spectrometers running. So this is a kind of very unusual technique to use in this space. So what these three traces that you can see on the screen show you is that the lower trace, the bottom one, is actually an extract of peat. So what we did is we just mixed peat with some methanol, which is an organic solvent, and we did some NMR spectroscopy of what we found. And we did the same with spent coffee grounds, coffee waste, and we did the same, just for the record, for decaffeinated coffee at the top. And you'll see there's a lot of signals. Each of these signals represents a series of chemical compounds. The phenolic region of the spectrum, where you'd expect to find phenols, is this region here where I'm circling. And you'll see in the peat spectrum, there are some signals down here. So it indicates that there are some phenolics, which is what we'd expect, of course. But remember, what we're doing here is we're just extracting chemicals from peat. We're not actually cracking it. We're not burning it. And therefore, the amount of compound that we're getting, the phenolic compounds that we're getting out of here, may not be particularly high, because in order to get those phenolics, we need to actually heat the peat up. So this extraction method that we're using here isn't maybe the most reliable or the most informative. And actually getting any kind of small molecules out of peat is very difficult, because it's such an intractable mixture. It's been called the most complex mixture on Earth. And so it's not surprising that we don't see a huge amount of signal down here. But you can see that there are signals in the coffee ground and in the decaffeinated coffee, also in regions that we would expect to find phenolic compounds. So there's some kind of encouraging news here that we're seeing some data that suggests that maybe we will be able to find these phenolics in these substitute materials. So what we can also do is we can actually burn peat and indeed coffee ground, and we can trap the chemical compounds that are produced in the smoke. And this is a kind of homemade bit of kit. So this is a commercial smoking gun that people buy and use in barbecues, for example. And there's a little hopper here that you can put peat or coffee or any other substitute. And if this is lit, so if you catch fire, then air gets blown through. It's a bit like a hairdryer, and you can blow smoke out through this black tube. And you can just about see there that we've got a little sample vial here where we're collecting the smoke. The smoke's bubbling through this solvent here, and any soluble molecules that are in the smoke will be trapped in this solvent, and we can do NMR. So it's kind of cool, because this is NMR of smoke, which isn't something that you normally expect to see NMR spectra of. And again, you can see that there's a series of signals, and now the peat at the bottom, the coffee in the middle, and the decaf coffee at the top. And you can see now there's starting to become a little bit more action in the in this peat spectrum, because we're cracking the peat now. We're actually burning it, so we're generating these smoky phenolics, and you can see that there's also evidence of them in the other two spectra as well. So maybe we're on the right lines here. Maybe we could get these compounds out of coffee if we kind of investigated it further. So this is another technique, and this is what's known as pyrolysis GCMS. So this is probably a more useful tool if we're doing this kind of analysis, because what this involves is actually burning or pyrolyzing, to be more accurate, the material that we're interested in. So this is heating up in the absence of oxygen. So what happens when you burn biopolymers is that they break up, they crack, They generate small fragments that come up in the smoke, and you can analyze these fragments using mass spec. So this is really what these two traces are. And you can see at the top here, we've got a sample of peat from the island of Islay off the west coast of Scotland. And you've got a lot of these signals here, and I've tried to label some of them, and all of these compounds that you can see labelled here are compounds that would be useful in the production of Scotch whisky, because they all have a flavour profile which ties in with these smoky notes that we're very interested in. And then if you go to further along the spectrum, then these are much higher mass chemicals, long chain hydrocarbons and things like that, which are probably not going to be influencing the flavour very much, because they're just simply not volatile enough. So most of the volatile compounds that we're interested in are on the left hand side of the spectrum. And you can see if we do the same trick with spent coffee ground, again we get a series of phenols that we're familiar with now from earlier slides, and again later on these much larger bulky long chain hydrocarbon compounds. And you'll notice also in the coffee ground that there are these fatty acids, palmitic acid, stearic acid, etc., coming off. And these are the lipid fraction of coffee, which isn't really present in peat. So we have this additional component in coffee ground that isn't present in peat. And these materials here, these fatty acids, are of quite significant interest as potential substitutes for palm oil. And so spent coffee ground has been looked at as a potential source of a substitute for palm oil, which again, rather like peat, is very much in the news at the moment in terms of its sustainability and the crisis that we're in terms of extracting that. And we can tabulate data using yet another technique. So this is what's known as SPME, or solid phase micro extraction mass spec. So what this involves is taking the malt that we've kilned, either with coffee ground or peat, extracting the chemical compounds from that, the small molecules, and we can identify and tabulate them. Again, these are lists of phenols which will be very interesting to us if we're trying to design substitutes for Scotch whisky. And you can see that there's phenols present in the lab peated, the industrially peated, so these are two different kinds of peated malt, and also the malt that's been treated by SCG. But if you look at the quantities down here, they're variable, so they're not the same ratio of compounds that you see, for example, in the peated malt. So the message that that's telling us is that although we have the same kinds of compounds coming off in the coffee ground compared to the peat, the ratios of those, proportions of those are different, and you'd expect that to impact on the flavour of the final Scotch whisky once we actually get the thing produced. This is a really interesting slide because it's back to NMR, and it's NMR of whisky. So whisky, kind of whisky called new make whisky. So this is whisky before it's matured. So according to the Scotch whisky regulations, in order for you to call a whisky Scotch whisky, it has to be matured in an oak cask for at least 3 years. Obviously, 3 years is a long time to wait when you're doing scientific experiments. And so we tend to use these so-called new make spirits rather than the final matured spirits. So these new make spirits are the spirits before maturation. It only takes about 2 weeks to make Scotch whisky new make, which is okay, but it takes 3 years to make matured whisky, which is obviously not very good for us if we're doing analytical science. Excuse me. And these three spectra here are a spectra of new make whisky spirit, and you can see that they are very similar. So the three different spirits, again, the two lower ones are these peated spirits, and the one at the top is a whisky that's made from spent coffee ground. So we actually made a whisky spirit using spent coffee ground rather than peat in the kiln, and you'll see that they look very, very similar. There are differences, so there are differences here. So these group of signals here aren't present in the peated spirits, and these signals here are also not present in the peated spirits. And what's interesting is that we know what these compounds are. And in this group of compounds here are nitrogen containing aromatic compounds, pyridines and pyrazines, and those pyridines, as anyone who's ever smelt pyridine might know, have a very unpleasant acrid smell. And so the presence of these compounds in the whiskey spirit is most undesirable, and I'll come back to that in a moment. This other group of compounds down here are furans, which are cyclic compounds that contain oxygen, and they're present usually in whiskey spirits, but not at this quite high concentration. And again, at high concentration, these furans deliver rather unpleasant aromas and taste. So we don't really want those in the whiskey spirit. So what we're able to do here is to try and tie in the chemical composition of the whiskey spirit with the sensory response. In other words, what it tastes and smells like, to develop a deeper understanding of what compounds we need to be present and what compounds we don't want to be present. And this is actually the key to all of this. So this is a sensory analysis slide, moving away from chemical analysis here. So this is a sensory analysis. So this involves the smelling of the whiskey spirits by experts. They are, once these spirits are nose, as the technical term is in the whiskey industry, then they are assigned a series of descriptors, burnt, smoky, and medicinal in this case, which describe the different elements of the flavor profile according to the views of the sensory panel. And you can see that there's a great deal of similarity with the spirits here. But the only one that's kind of well out of kilter is this 1.21 here in the case of the coffee ground, which is significantly higher than the laboratory and industrially peated. Now, you don't really need to know what these numbers mean. All you should really understand is that there's a big difference here compared with the numbers in the other columns. And that reflects this extra burnt, rather acrid flavor that comes from the presence of these pyridines and furans in the coffee spirit that isn't present in the peated spirit. So what we've been able to do here is to map a sensory response, which is a kind of extra burnt, almost like a burnt plastic kind of aroma, to specific chemical species within the whiskey spirits, which is kind of funky. This is really the last kind of analysis that I want to show you. And it's a technique called ion cyclotron resonance, a mass spec, ICRF-TMS. And this is a remarkable technique and has almost unmanageable, unbelievable sensitivity at picking up traces of many compounds. So this is a technique which uses a cyclotron, which is a kind of particle accelerator, to separate chemical species before they're analyzed by mass spec detectors. And it can identify huge numbers of small molecules. You'll see that this is what we call a Van Crevelin plot, and it's a rather unusual way of displaying data. Because we get so much data from ICRF-TMS, we can't really plot it in the way that we plot normal spectra, the ones that we've seen already. We actually have to kind of bundle the data together. So what we use are these ratios of hydrogen versus carbon against oxygen versus carbon, and we get almost like a kind of imaging map, which is the Van Crevelin plot. And each of the dots here represents a specific chemical species. And because we know that certain chemical groups will appear in certain regions of the spectrum, for example, lipids over here, carbohydrates over here, lignins over here, we can get an overview of the chemical composition of the whiskey spirit in a way that no other technique can really deliver. And you can see on the right-hand side here that we've identified almost 4,000 specific individual chemical compounds in the coffee waste whiskey using ICRF-TMS, which is even more than you'd get in peated whiskey. In peated whiskey, you normally expect to see 2,000 to 2,500 identifiable compounds. So the whiskey spirit is incredibly rich in small molecules. So we need to do some much more work now in trying to identify what these are. So the kind of conclusions to the to this part of the talk, I won't go through all of the conclusions. You can see that that they're on the slide here. But I think that the key point to bear in mind is we can use analytical science in all kinds of spaces, even in spaces that you wouldn't imagine, like in food and drink, for example. And we can use traditional and new analytical science techniques to solve challenges, to understand problems and then try to find solutions to them, and around climate change, sustainability, renewability, energy, food and drink, and all of these different areas that we're also interested in at the moment. I just want to spend a couple of minutes now talking about the program itself, and there'll be an opportunity to ask questions afterwards if anyone has any, and of course you can contact me directly if you want to. So the full title of the program is Analytical Science for the Biosciences Industries, and in terms of what we mean by biosciences, biotechnology, brewing and distilling, which is kind of in the space that we've just discussed with the whiskey project, food and drink, also the pharmaceuticals, obviously the production of drugs, and clinical products, bioengineering, also biomanufacturing, so that increasingly bugs are being used to make chemicals and drug compounds rather than traditional chemical methods, production of agrochemicals and that kind of thing. So it's a very, very large area encompassing a very, very significant number of big industries. And one of the things that we've discovered through conversations with our industrial friends is that there are skills gaps in traditional analytical science. In other words, people that want to move into working in these areas in analytical science labs really do need to have a full knowledge and toolkit of analytical science, the techniques and technologies involved in order to be able to operate effectively in that area. And so this program is really designed to give you those experiences in order for you to move into that sector, if you so choose, at the end of the program, and be able to operate very, very effectively from day one. The other interesting thing about the program is that it doesn't just tell you about techniques and technologies and science. It also gives training on what we sometimes call softer skills, which is a little bit insulting. So these are skills around health and safety management, project management, regulation, which is key in biosciences, particularly if in the life sciences. So all of these life sciences labs operate under strict regulatory regimes, which can become something of a shock for people who haven't actually worked in that space before, who come from universities and schools that haven't had to deal with those kind of regulatory issues. So we also give a lot of information on that also. And so we came up with the idea of designing this program, and one of the selling points of the program is the fact that we collaborate very closely with industries to help us develop content to make the program relevant. So I've listed some of them here. There are many others, and we have frequent conversations and collaborations with our industrial friends that guide us to the kind of content that we should be delivering to make it as relevant as possible to the students, and that obviously improves employability once they've finished the program. I won't read this all out. You'll get the opportunity, I think you'll get copies of this recording, so you'll be able to read all of these slides in detail. But we have one of our students from last year, Lee McMahon, who's actually stayed at the university to do a PhD in chemistry and in education-related PhD, actually. So you don't need to carry on and do analytical science. You can use the skills that the program give you to move into other areas. And I think Lee was particularly interested in the fact that it was a very interdisciplinary program with elements of chemistry and biology, technology, instrument design, that kind of thing. And also the connection with industry, as I mentioned earlier, and also the ability to network and meet potential employers through these industrial collaborations and projects. So I think these are the key things that Lee really appreciated about the program, and Lee did very well, and he graduated with a distinction from his program. So we have close ties to industry. The projects are all inspired by industrial real world problems and all have support from industry. And the kind of projects that we run are all about teamwork, solving problems, being able to present your results, dissertation, and in a presentation, all these associated skills which are so important if you're going to go out into the real world and work in the analytical science space. I think that's everything I have to say. So thanks very much for listening, and I will close now. Thanks very much.